Today we are going to discuss about a type of density gradient centrifugation known as isopycnic centrifugation. Okay. So isopycnic centrifugation, also known as Bowen density centrifugation, also known as equilibrium density centrifugation. So isopycnic centrifugation separates material based on their differences in Bowen density. Okay, so what is the meaning of the Bowen density? I hope you know. The Bowen density is the effective density of the macromolecule suspended in a liquid. Okay, that means it is a relative term depending on the type of the medium in which it is either reside or through which it is sedimented. Okay, so the main funda of isopycnic centrifugation is same. Isopycnic centrifugation separates on the basis of their differences in density, right? So isopycnic centrifugation separates molecule on the basis of their differences in density. So what is the idea? So first of all, here we will take a tube, that is centrifuge tube, and in the centrifuge tube we will take the matrix forming substances. Okay, the matrix forming substances. So these matrix forming substances have some special quality. What kind of quality? First of all, these matrix forming substances will generate a gradient. Okay, gradient means there will be a continuous change in something throughout the column. Okay, so here the gradient is of density. That means the density will be continually changing from the top to bottom. Okay, so another important property of that isopycnic centrifugation that I want to mention, that is this matrix forming substances that we are using not only produce density gradient, but these have another distinguishable property, that is it generates the gradient after the completion of the centrifugation process. Okay, that you do not find in case of another density gradient centrifugation such as red zonal centrifugation, such as sucrose density gradient centrifugation. That is the idea, that in isopycnic centrifugation the gradient is not formed in the first place. You will have your gradient differences only after the centrifugation. Okay, so we have taken a matrix forming substances in a centrifuge tube. The second step and the second choice of us will be a uniform mixture of the sample that we want to separate. Okay, here the term uniform is very important. It is the basic requirement for any isopycnic centrifugation. The solution should be uniform. Okay, so we will mix those uniform substance to be separated with the matrix forming substance in such a way that together the matrix forming solvent and the solute mix together and leads to the formation of a homogeneous mixture. Okay, so that's what I have written here, that in isopycnic centrifugation the next thing we need is the unique mixture of sample. That is very important. Unique, or you can say uniform, it is the same thing. The sample is added into the column and mixed in such a way that it will give us a homogeneous mixture. Okay, with the matrix forming substrate. So then after the formation of those homogeneous mixture, we will load them in a centrifugation machine, that is known as centrifuge. Okay, and the centrifuge will spin for a certain period of time. Okay, and where the molecules will move about an axis, and that resulting in the perpendicular force called centrifugal force that we have discussed, on the basis of which centrifugation is done, right, and it will go on for up to certain time, then centrifugation will stop, and after centrifugation we will end up with a gradient. Okay, so that is the idea. After centrifugation we will end up with a gradient that is not formed prior the centrifugation. So now I want to discuss with you about what is the basis of that test. So the A test tube is the condition prior centrifugation and B test tube is the condition after centrifugation. Okay, so the basis of the separation or gradient formation is nothing but the Bion density of the particles. Okay, so here the macromolecules will pass through a column whose density is adjusted, right? Here the lowest density will be at the top and the highest density will be at the bottom. Okay, and the intermediate part will take the medium density. Okay, and through which the macromolecule will move and they will settle at a particular point where their Bowen density will be exactly equal to the density of the column. Okay, that is just like the separation in case of your isoelectric focusing, where when the protein pi value match with the pH of the certain environment, it stops at that point, right? So similar thing is going on in case of isopycnic centrifugation, in which the macromolecule will stop sedimenting when its Bowen density will match the density of the particular position of the column. Okay, and as a result we will get distinct band. And each of the band will contain different particles whose Bowen density is exactly the same. Okay, that's how with the basis of the Bowen density we are separating a complex mixture with the help of isopycnic centrifugation. Okay, so I am repeating the entire thing. The first goal of us to take a matrix forming substrate in a centrifugal tube. Okay, now this matrix forming substrate have some special quality. So this matrix forming substrate will produce gradient. Not only that, this gradient will be generated after the centrifugation. That means we are not going to have the gradient at the first place. Okay, so after that our second requirement is a uniform mixture of the solvent, uniform mixture of the sample to be separated. Okay, and then we will mix those uniform mixture of the sample to be separated with the matrix forming substrate, and they will generate a homogeneous mixture. Okay, then this homogeneous mixture containing tube will be spun in a centrifuge. After a certain time the centrifugation will be completed and we will get distinct bands that we call gradients, right? And it is totally based on the Bowen density. When the macromolecule's Bowen density matches the density of the local environment, it stops at that position. Okay, and it is totally time independent. And another important thing that I need to mention, that in isopycnic centrifugation you do not get any molecule that settles down at the bottom. As you can see, there is no molecule that settles at the bottom. That is the another interesting thing that I want to mention to you of isopycnic centrifugation. Okay, so that is the completion of the topic, and I am wanting to explain another important thing, that what are the different properties that we should keep in our mind when we choose the matrix forming substrates. So first thing is, matrix forming substrate will be cost effective. Okay, and it should be easily autoclavable, right? And another important thing, it can be easily separated from the samples, right? And the matrix forming substrates cannot be metabolized by the samples, right? Because sometimes we are separating cells also, so we have to keep in your mind, in this thing in our mind, right? So another important thing is the matrix forming substrate should not be toxic to our sample, and it do not react with the biological molecules. Okay, that are the various important points that you have to keep in your mind during the choice of the matrix forming substrates during the isopycnic centrifugation. And what is the difference of isopycnic centrifugation with the other type of density gradient centrifugation? The difference is, in case of isopycnic centrifugation, you will get the gradient after centrifugation. Okay, on the other hand, in case of zonal centrifugation or sucrose gradient centrifugation, you will get the gradient in the starting of centrifugation. That is the difference. Okay, so that is the idea. Here at one tube we will keep our sample, another one will be balanced, and it will spun to generate the centrifugal force, and as a result we will separate them, okay, in gradient. Okay, so I hope you get that. Now I am just go through the slides so that you can take screenshot for your own purpose. Okay, so that's it for today, friends. So today we have discussed another kind of density gradient centrifugation, that is isopycnic centrifugation. In the next video I am going to discuss, or I should say I am going to recapitulate some important concept of basic concept of centrifugation. Along with I will also try to complete the differential centrifugation. Okay, and we will left with just one topic more, that is red zonal centrifugation, that we are going to cover in the third video that I am going to upload regarding the centrifugation. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. And thank you for listening to my class.